So I hope it kind of interacts with some of your interest. As Max says, one of the main aims, like explicit, if you want, non-epistemic aim of this project, in a way, is to contribute to stop racism in science and society. And this is because just science is such a powerful thought machine producer that like it really has an effect. So in a way, you have to kind of, this is like a framework where this research comes from. This is the project I was talking about. It's called Microbiome Research and Race in the Local South. And the idea of the Local South is to try to pay attention to how research is done locally, not only in terms of what kind of questions, more like in this epistemic sense, but also what kind of conditions, limitations, or structural inequalities are found in different locations in the world. So we look at microbiome research done in mainly in South Africa and also in Colombia, Mexico, and Uruguay, some places in Latin America. And what we want to do is to identify population descriptors or labels, generate racialized views of populations, or create harmful stereotypes, so that besides labeling people in their studies are also kind of having an impact on how these populations are perceived. And as I hope I can convince you, this impact is not only in society, it's also in science. Now it kind of reproduces itself through different ways of framing these populations. And we also would like to generate new and more accurate population descriptors, because it's a problem, right? You want to describe the populations you're working on, so you really need to label them somehow. But maybe the question is, which kind of descriptor then is very suited in different cases? So this is people who I work with, and Utrecht is not that far away in case you wanna visit us, and it's also nice when the weather is better. So today I will talk a little bit first about race and introduce some distinctions, just like race, racism, and racialization, and why I think it matters to have different words or different concepts to name things that we see happening. And then I will introduce you more to this idea, or like this problem of using race or ethnicity in microbiome research in connection to health. And at the end, I will talk about what we're trying to do. So this project is 5 years long. We're in the half. So I don't have all the answers here. We're just like kind of in the process of bringing new methods in order to possibly get the answers in hopefully 2.5 years. So that's what I'm going to talk about at the end of the presentation. So just to start with a very basic introduction about race. I'm pretty sure you know about this, but so research in the history of race, or history of science and history of racial science, has shown that scientists have focused across time on different body parts to define racial categories. And they have changed from, I don't know, the skull to different phenotypes, to very scientific ways of measuring people, to different devices to also very objectively kind of gather how people are different from each other. So in the history of racial science, we see a lot of effort in really kind of making kind of objective judgments of how people are different. This has, of course, failed, but we have several examples of how racial categories have been trying to connect to something in the body. Now, it can be blood, genes, skulls, face shapes, etc. So in the historiography, or the traditional historiography of race in science, one of the first moments where the concept of race as something that is just used in science but might be problematic comes after the Second World War. And at the time, so kind of organizations like UNESCO decide to, talk about race, because it's a concept that is broadly used in science, but it's very problematic, right? So they write a declaration on race that in these years where different scientists, natural scientists and social scientists come together to talk about this concept. At the time, they decided that actually race is a biologically valid category that they need to use in order to do studies on people. However, that doesn't necessarily justify to have racism in science. So it doesn't justify to create hierarchies among different human groups. So they kind of go in favor of an anti-racist science. So this consensus eventually changed, and scientists start denying the validity of race as a biological category. And I think, well, I don't know if recent developments, but at least at some point, biologists and scientists were more or less on the same page on saying that race is not necessarily a biological category, is rather a historical construct that has shaped society in many important ways. What happened, what some historians of science had also shown with their important work, is that in a way, this replacement of race as a valid category towards the use of population in many different biomedical studies, which is something we find in a lot of research today, the use of population, happened rather in a way, in a kind of an implicit way. It was substituted little by little without doing like make it very explicit. And it was also like part of the popularization of population genetics at the time, not like at the 70s, 80s in biology. So there's kind of a process where race starts in decline to be used in science. Population is kind of substituting the use of race, but still there's like things that are unspoken in the way that, well, you still have this idea of we need to group people in something that was called races back then, and now we're calling populations. We don't use the word, but what are we doing with these groupings? No, is it like only a conceptual substitution, or rather a different way of understanding populations? So that's kind of based on the work of colleagues from us who go to history of science, and this is kind of a really very short story about how that happened. So then kind of the second moment where people say like, okay, now really race is gone, is with the Human Genome Project in the 2000s. And what people thought back then is like, okay, now it's clear that we're 99.9% the same. You know, that's what apparently the Human Genome Project showed. So there's no way we can be different races, no? If we are so similar, where can this idea of biological race be? So that was kind of the second moment, and it was believed to be the moment where finally we're going to close the door to the idea of biological race. But then very, very soon, the focus on sameness, the 99.9%, changed to the focus of difference, where 0.1 different, and that's what matters. So let's take a look at that. And this, to understand better how this focus towards difference happen, I think it's really interesting to look at, for example, research done by Stephen Epstein on what he calls the diversity and inclusion paradigm, which is actually quite central to biomedicine. So in this book, which is a very good read, I can recommend. What he's describing is a situation where previous to, like, let's say, like in the 80s and so, the understanding of human diversity was, or like the way the research was done in biomedicine was focusing on gathering groups of white males, young white males, and kind of extrapolating these results to other population groups. It was, of course, wrong. No, we know the problems of doing that. And it was only until the 80s that kind of people start reclaiming or like asking for more diversity in biomedicine, which is really important. Kind of the problem is then in which way and in which context and under which framework you are then creating diversity. So this is a quote from this book, The Orthodoxy of Sameness, which was before this diversity and inclusion paradigm happened. what has dominated much of the thinking in medical science, often impaired our attitude toward clinical research in those days. So the idea, as I said, now there was like this paradigm of, oh, let's just look at this, like to reduce the human to the 60-kilogram white male, 35 years of age, and so on. So it's actually, diversity is really important because then you can get results for different populations. The problem is like how you name these populations. And this leads us to the second kind of part of the story, which is that when, as you might know, the United States are quite a very big, powerful scientific pole that generates a lot of funding and research. And the fact that these questions about the human genome sequencing and the questions on diversity kind of emerge in this context also flavor the way we understand the diversity in terms of racial diversity as they use it. So I don't know if you have, are you from the US, anyone? Have you gone to the US in the past? Have you had to cross the little box that you have to say whether you're Caucasian or Afro-American, or, I mean, I think I would have to say something like Hispanics or Latin, whatever, but I feel very uncomfortable doing that. My point is that in the US, the structure, racial categories are at the base of their administrative processes, and everyone has a race, right? So what happened then is that when this kind of framework enters biomedicine, it will bring with it these distinctions and this view on diversity, which is what I think we are seeing in biomedical research today, or at least parts of it. So, so that's kind of, that's why we have population descriptors. That's why we have labels in biomedicine. We need them. We need to label the groups. We want biomedicine to bring benefits to everyone, to all populations, hopefully. And for that purpose, we need them to go to study different populations, and we need to label them. So the scientific rationale for describing research participants and their group labels is what is defined as a population descriptor. These descriptors are supposed to represent a concept of difference across human populations, and this concept of difference needs to be relevant, right? You can label a person in many different ways. For example, me wearing green, and maybe you and me, we can be a set of people today. That's probably irrelevant for our health status. So kind of the question is how to find the relevant ways in which you can group people under population descriptors that make sense for the question you're asking. The kind of population descriptors that are more popular in biomedicine, as you might have seen, is, or not, I don't know, race, geography, ethnicity, gender, age, ancestry. So these are the kind of differences that are important to know in case you're going to do like a study or something like that. And as I said, this is assumed when these population descriptors are used, and when different studies compare or contrast human groups that are labeled with some of these population descriptors, the assumption, as I said before, is that these reveal meaningful biological differences that are relevant for health. So it's not just like any other difference. It has to be something that is relevant for health, and it's biological. And that's kind of where my concern for the use of race, ethnicity, and other descriptors that I will show later comes in, because I see that a lot of this research are still kind of describing a lot of these ideas of race and different racializing views in the way they're labeling populations. And before I continue, I just wanted to bring some definitions, because I think when we have words to talk about things, if we don't have words to talk about things, we cannot name things the way they are. So I usually like to differentiate between the concept of race, which I describe as a system of human classification established historically in relations of domination to naturalize the belief on intrinsic human difference and hierarchy. So it's kind of something with the purpose of kind of creating this distinctions in humans, where white people are superior physically or biologically, intellectually, and aesthetically, they're more beautiful, and culturally to non-white people. Now, and this is something that we see, of course, developing across many years with different shapes, different ways of relating, but in general, we see a kind of a white supremacy idea in how different populations are different or the same. So it's pervasive, historically and geographically contingent. Then I think we also have to separate that from racialization, which is much more like a process. So when we're talking about race, we're talking about things like groups. Racialization is something you do to something else. And I like the definition that Adam Hochman has of racialization as a process through which racialized groups, rather than races, so you kind of racialize a group, are formed. So when we are actually applying a racial category to a group, we are racializing them, but they're not necessarily a race, right? But talking about racialization has some sort of also a value, Hochman argues, and I think he might be right, as it offers a way for groups that have been understood and treated as inferior races to assert and define themselves collectively while rejecting the biologization and inferiorization associated with race. So basically you can say, hey, I've been racialized for, I don't know, my life, but that doesn't mean that I belong to a race. That's something you're doing to me. And then in that sense, you can also talk about the problems that this racialization has caused to you. So it kind of allows to talk about it in a processual way. So, and also the benefit is that it goes beyond explicit race labels, because as we will see, racialization can happen also when race is absent from discourse, which is kind of something that we will see very often also in microbiome research. And lastly, I have also the definition of racism, which is different from the previous two. And for this definition, I'm relying on the recent book of Peter Wade and Monica Moreno on racism in Latin America. And what they say is that racism combines basically two elements. One is a historical origin in European processes of reconquest and rechristianization within Europe. And I think I like this definition because it's bringing religion to the mix, in the sense also telling us or reminding us how these racialization processes are not only biological, they are kind of going into much different, like in different levels. So expelling Muslims, Jews, forcing conversion and discriminations, goes along creation of Western modernity, through metrics of color and geography. Hi, welcome. And the second element from that they identify is that racism has this discourse of natural cultural that links bodies, behaviors, and biocultural heredity. It is transgenerational, fixed link, and is passed by blood, genes, etc. So when you are kind of part of subject of racism or part of a racialized group, this will something that you can inherit to someone else. Of course, this is also historically contingent and change with geography. What they also emphasize is that racism, just as race, is kind of some sort of creation to distribute power, resources, privilege unequally through violence and hierarchy of value. And then reveals also some sort of grammar, affective and logical of naturalization that maintains unequal distribution. And I like this part of the definition because I think something really interesting about race, racism, and racialization is that it's not only logical as something we think, it involves the emotions of people. It involves a sort of rejection of the other in a very visceral way. And this is something that I think if we don't acknowledge, also when we are talking about science and scientific discourse, then it's kind of, we cannot disentangle really what is happening when we read all these racist things happening currently. So those are my definition parts. I hope we can discuss them later. Now I'm going to move into the microbiome. Do you know what the microbiome is? Yeah. Okay. So the microbiome is amazing. It is, I mean, when people discovered that we had so many cells, like much more cells or genes than human genes, it was kind of like, what? We thought we were humans, right? Well, no, we're kind of like a collective of millions of bacteria, viruses, archaea, fungi, etc., together with humans. And I think these numbers are not necessarily super updated, but they're kind of, I guess, not that different. But for example, I think this is, for example, very interesting. The genes in your microbiome outnumber the genes in our genome by about 150 to 1. So these kind of magnitudes of how we are basically a collective or an ecological system of microbial systems and human cells kind of puts into perspective what kind of information we were able to know, for example, with the genome, with the Human Genome Project, when we had so many other things going on in our bodies. So then we have microbiome in our gut, the skin, basically in all our cavities. And kind of the important thing of this is that it seems to have a very important role in our health and also in our normal development. So kind of the question is, how can we understand the causality between the microbiome composition and health and disease states? No? That's what scientists wanted to know. In contrast also to genes, for example, and let's leave it like that. In contrast to genes, microbiome is supposed to also change with your activities, no? So if you, for example, start eating completely different or start drinking coffee or stop drinking coffee, the idea is that these changes will be noticeable in your microbiome, something that was not happening, for example, with the genes. So kind of in a way, it really changes the way we can understand our porosity to the environment and how also we can be a bit of an agent changing our own microbiome. This is an illustration from a publication on gut microbiome research, which is the one in the gut. And it's just to show how kind of the framework that is behind this kind of research. For these researchers, the way we, as I said, our lifestyle, our food and our environment will have a very big impact on the microbiome. And usually what they think is that we can find at least three different compositions of the microbiome divided into subsistence strategy, which are hunter-gatherer, agriculturalist, and industrialized. Each of them have different ways of life, has different like settlements and food, but it's also, they have, at least what they're trying to do is to identify what kind of taxa are associated. So what kind of taxa are more dominant in each of these stages, and then they have, for example, Merotella and so on. And they tried also to identify what kind of functional features these taxa have in each of these systems, and also the relative diversity that this different microbiome has. The assumption is that the highest diversity is better than the lowest diversity. And as a conclusion, people living in industrialized settings, so basically cities, etc., are kind of in need of gaining again the diversity they lost. So kind of that's the narrative that's going on. I'm going back to this in a bit. So in a way, what we find in microbiome research is a combination also when we're talking about race between these kind of categories like hunter-gatherer, industrialized, Western, together with Latino, Hispanic, Western, Afro-American, etc. So there's kind of like a mix of different labels going on. And as I said, what is important here is that this understanding of difference, which to me, it could be race, but it can be also a racializing way of understanding differences, is taken to be a risk factor linked to health differences between the global north and the global south. As I said, usually industrialized countries, they're supposed to suffer from all these metabolic diseases and non-communicable diseases that come with their lifestyle. And people in the global south, which basically is, I mean, these distinctions are not really clear, but basically all those people that live like not in cities, but like more agricultural, pastoralist way, they're supposed to have much more richer microbiome, and then they're supposed to also have different health and disease problems. But in general, the kind of international group of researchers doing gut microbiome research are very much interested in looking at this kind of metabolic and more like global north diseases, which are obesity or cancer, type 2 diabetes, asthma, and other neuropsychiatric disorders. So, yeah. So, but I'm interested in the opposite, because the problem is that if you assume that all the industrialized people that live in cities are sick of the same because they have lost their microbiome, then what you do is to go to the others, to those people who are not living in the cities and they are living like in other situations, to find the solution to the problems of the people of the north. And this is what is happening currently. A lot of research is done on different populations, study, for example, in Latin America, in Africa in general, or in Asia, and look places outside the city to look for the answers for the different health problems that people in the cities have. So to look at this problem, and also because, as I said, I'm interested in how these different scientists or like these different publications are using racial, ethnicity, or other labels to describe people. What we did in 2021 was to do a literature review, not an extensive one, but like looking at gut microbiome science done in Latin America. What we wanted to understand is why were people going there? Our hunch was that it was for searching for these answers, but we wanted to look what were the justifications that they had. And what we found in these different studies was that Latin America, by the way, I'm from Mexico originally, that's why I'm interested in Latin America, just in case you were wondering. In Latin America, what we found is that scientists perceive Latin America as kind of an ideal playground because it has a lot of people living in non-Western lifestyles, which is basically meaning not in big cities or something, which is also kind of weird because Mexico City has like 3 million people or so. So anyways, or like Sao Paulo, home to non-Western lifestyles. So basically meaning uncontacted hunter-gatherer populations. They also think that Latin America is a place where you find a huge diversity of host genetics, and this is related to how we imagine the origin of Latin American populations as the result of colonialism. So there were like the natives and the Spanish colonizers came, and then came the slaves, and that's like an hybrid set of genetics going on that you allegedly don't find in other places. And also the problem is that a lot of Latin American populations are have chronic diseases, are sick. So you want to know why they are sick, but still they're like so rich in genetics and they live so differently. And this is why going to Latin America is basically having access to health relevant ethnic and racial diversity and also their microbiomes. So when we did the study, we found out at least two narratives that we thought it was interesting to see. One of them is that people in Latin America, so basically deprived indigenous and mestizo bodies. Mestizo means a mix, what I just said about Europeans and stuff. In developing countries or with traditional lifestyles have become sick and at risk. So either Latin Americans are framed like this, like you're living in a deprived environment, and this is why you're getting sick, etc. Or a narrative about the purity and primitivity of uncontacted indigenous groups that are decoupled from a Western lifestyle. So a very idealized way of understanding people who are living, for example, in Amazonas or places like that, and are allegedly uncontacted from civilization. And these two narratives to us, according to what we did, seem to be really relevant as in guiding researchers to go to specific places to do a specific research. So it's kind of the justification of why it is important to do the work they're doing. And I'm going to talk about only about the second, about the purity and primitivity, specifically related to this group of people who are the Yanomami, who are living in the Amazons for, they're living there. And they have been, because of the way they live, they have been studied like for at least 80 years. Anthropologists have been there often. James Neal, for example, who was famous for his theory of the thrifty gene, if you ever heard about that. Went to them also to understand, how they behave, etc. And believe, for example, that the world of the primitive man is remarkably uncontaminated. He went to many different places, and this was one of his conclusions. But what is interesting is that still in 2015, so some years ago, and still quite often cited, microbiome researchers frame this population as uncontacted, as for example in this paper, the microbiome of uncontacted Amerindians. So the problem is that, well, in this paper, current microbiome, these authors argue that the Yanomami is an uncontacted community that represents a unique proxy for the pre-antibiotic era human race system. So some sort of community that has never or has little contact with antibiotics, which is some of the things that can really alter your microbiome. But the problem is that by this claim, they are completely forgetting when they got contaminated by different things, and they actually needed antibiotics. So they have this community is not uncontacted, and it's not pre-antibiotic. It's just like a kind of a proxy of this. Another argument in this text is that Yanomami carry a pristine bacterial gut, skin, and oral diversity. And the understanding of this microbiome can reverse the current global trends in metabolic and inflammatory diseases of the industrialized people. So kind of, in a way, the purpose of going to this population is not necessarily to know about their microbiome for the sake of knowing about their microbiome. It's because we want to know something about how to fix diseases from other people. So in some way, I think that this kind of framing is treating these populations as primitive, decoupled from Western lifestyle and frozen in evolutionary time. And this is, of course, not only racializing, but it's also producing racist ways of looking at these people. And it's also somehow, it's not true. This population is not an example of a pre-era resistance. So it's epistemically wrong also to assume that this population represents that. What is more is that, okay, you can say, okay, this is a paper, 2015. Yeah, but the problem is that these papers are quoted and quoted again. And this framework is really at the core of how microbiome research is done, assuming that there's still a group of populations out there that are somehow isolated and somehow very different than the industrialized populations. So I'm not saying that there are no differences. Of course, there are differences. The problem is which differences and how do you find the relevant differences to know what is important for the microbiome for it. This is another representation of what microbiome research framework of change. Just as the other one, it's also quite common to find this quotation. And I just think it kind of represents the other image I had with the 3 ways of living in different ways, and also with kind of more like a movement arrow where microbial diversity decreases when you move from the hunter-gatherer state to the industrialized, Western, urbanized. And also, but also I think it's important to see here that it says Neolithic revolution. So in a way, it's also kind of framing this other group as being pre-Neolithic revolution as if it was in the past. And as I said again, okay, this is a paper, whatever, but the problem is that these papers are more and more like all the time that referred to as a framework where you have to base your research from. So it's something that still remains at the core of how scientists are kind of thinking about what they have to do research about. So kind of what I want to, the point I want to do here is that it is not only an ethical problem or a racist problem or racism or something that had be or something moral. It is an epistemic problem what is happening here. If at the core you have assumptions that are wrong, you cannot get research that is right. And of course, you're not going to get also the health benefits that are promised. So this is something that we're trying also to discuss with microbiome researchers. So to see how race or ethics are not like something additional. They're something central. And if you, and these, getting these things wrong, these are kind of ethically wrong things, can also impact the results of your research. So they're not decoupled. So as I said before, this narrative about purity and primitivity assumes, for example, that these non-Western indigenous, the uncontacted, are healthier than urbanized Western lifestyles. And this is why researchers go to collect these microbes of the communities before they disappear, because of course they know that, you know, the cities are growing, these traditional ways of living are getting lost, so we have to collect these microbes before they're gone. They're also doing this because they hope that these are key to the health of the people in the cities or in the global north. So basically, we see a dynamic where global north research questions are solved with global south samples, which leads also to stereotypes of indigenous population and extractive sampling practices, and reduces health to microbes while destroying livelihoods. So we also think that it's not only about the populations, but this kind of this logic of, oh, we collect microbiomes because they're going to disappear, is disregarding completely the whole set of knowledge and practices that precisely made these microbiomes possible. As I said before, lifestyle is what gives you your microbes. So if these people are going to disappear, I mean, if you just want to collect these, nothing warrants that you're going to get these microbes to persist because the lifestyle is gone. So kind of the emphasis is completely wrong. And it's also a very reductionist way of understanding health, right? Like, because it's not only the microbes. So kind of the question that we also are trying to understand then is who determines the focus of the agenda. I think you already know where I'm pointing to, but we think that there's a very big bias in how global health works and also microbiome research. These numbers are from genetics, but similar things happen also in this field. So biomedical research is largely shaped by global north agendas, with 85% of US studies funded by, for example, the US, well, at least until recently. I don't know, a new world is coming, I don't know, but anyways, 72% of the samples are European ancestry. So there's like, you know, everyone is aware that there's some sort of a gap and there's overrepresentation of specific populations. And this underrepresentation is something that is also worrying for microbiome researchers. However, we also think that this logic of completeness and underrepresentations serves the purpose of high-income countries and is racialized. For the same purpose, same reason that I said at the beginning, when you are trying to have a complete database, but your notion of diversity is racial, then you're racializing those who you're collecting. So kind of the framework in itself is somehow generating continually the racialization of people. And also, it is very, very useful to claim that your database is not complete, because then you can say, I need to sample more. I need to go even to more isolated populations, and I need to have everything all around the world. So kind of really motivates this act of sampling. So data completeness became, in a way, the operationalized counterpart to a very specific understanding of diversity and inclusion. So I said before, this is very, I mean, yes, we need diversity in databases, but in which terms, no? And also we see, if you look at history of science, we also see that global health institutions do have a very close connection, or like a historical connection, to European colonization, as for example, in the shape of colonial medicine, missionary medicine, tropical medicine, and international health. So basically these institutions that were put in place in different colonies to take care of the health of those people kind of developed into institutions of global health. So the question is also how global is global health? And for example, when we see that local diseases are underfunded, even if they are also suffered by a huge number of people. And well, this has also been part of the research that historians of science do on institutions and global health. There's also, in some people more working from the colonial studies, have also looked at the concept of global health and how it works in these institutions. They have found supremacist ways of seeing, that is that in many cases, headquarters that are located in New York, or I don't know, are taken more seriously than regional and country offices, who might be more informed about the local challenges that people suffer. There's a lot of focus on national governments instead of subnational governments. So again, the global instead of the local. And there's a greater value on research by high-income countries or distant experts than the knowledge of those with lived experiences. And in a way, what we're trying to also kind of put together here is, again, to rethink in which way, what is what matters here? I mean, what is the question you want to answer? If you want to answer that question on health, and as in my previous example of the microbes and the ways of living, perhaps the microbes are not the answer, but other kind of collection, other kind of knowledge needs to be understood. If you want to just like have a very big database, then perhaps you just have to continue doing these collecting practices. So kind of the question is who is kind of imagining what well-being is, in the sense of this desire of being healthy, and how is this put into work by these institutions. And I'm going to my last part of the talk. As I said, I don't have an answer, but I hope we can discuss that later. So what we're doing is to try to discuss about these problems with microbiome researchers. So I describe myself as a historian and a philosopher of biology, but what we're trying to do is to kind of do a little bit more of participatory for biology. We don't necessarily want to tell them what they have to do. We want to think together in order to find ways that are helpful for them to kind of question this framework that I was showing before about the hunter-gatherer and the transition and kind of these assumptions that are behind. So what we are doing.
So who is them?
Who is them? Microbiologists.
Okay.
Yeah, we work with, so we have collaborators, thank you. We had collaborators in South Africa, specifically in Stellenbosch and Johannesburg, in different institutes. And as I said, in Latin America, through the Latin Biora Consortium, who we talk constantly and who seem to be interested in that problem. And what we did, this is part of what we're trying to do to develop these conversations. This is a poster we did in our workshop that is not AI generated, it's generated by a person in this workshop in South Africa. So what we're trying to do is to start local dialogue. So we went to South Africa to do a workshop in which our main objective was that people could know each other. And there we organized a World Cafe. Have you been in a World Cafe? No? Okay, a World Cafe is like pretend you're in a cafe, but like with different people in different tables. And then you answer questions and people share and stuff. No? So the idea of those activities is not necessarily data collection, it's rather just talking to another people. And what we're trying to do then is to put together people from a specific region, so South Africa, Cape Town, and a microbiologist talking to a philosopher about race. And really exciting things happen, such as build an African empire that takes ownership of our own community. You know, like, let's stop following the North, let's be ourselves. Kind of really, I think, conversations that don't necessarily happen all the time. So what we were trying to do is to start these dialogues, is kind of just get to talk each other, get to become a community, know what kind of problems you have. And we're just talking about the scientist level. We're not going to the communities even, but I think that's more complicated, but like kind of talking with the scientists, because it's them who are creating these labels, right? So we want to talk with them. We're trying also to bring kind of a reflection on how to promote and develop own research agendas. And some of the topics that came in this workshop, for example, is that this dependency or the reliance on the US and Europe is also part of technology and economic gaps. So you just don't get this reactor, this liquid to do the DNA thing. You don't get it, it's very expensive. So you have to send your samples and then they do an embargo of 2 years and you cannot have them. And so it's kind of like a logic, also a dependency that is not only necessarily that is rather like a funding and technology gap, but that is necessarily shaping what kind of questions you can ask. So it's really having an impact in the kind of research and that you can do. Also, we're trying to see ways to understand health and well-being in plural. I think this is not something that we're inventing. There's a lot of philosophical debates on what is health and so on. But especially when we're talking about microbiome and when it's involving lifestyle, culture, etc., this is even more complex, and it's more important to acknowledge that not every idea of health is going to be the same. And this comes together to recovery of local knowledge and livelihoods. And this is also something that we don't necessarily need to idealize as, oh yeah, let's keep the traditional knowledge, from the Aztecs or whatever. Livelihoods and local knowledge is also what we do here, right? It's like, it's everywhere. It's the way we go about doing things. So that is what is important. What of the learnings that you got from your grandmother, from your mother are useful for health? So let's not forget about that. More in the conceptual part and about race, just to make awareness of race not being a global applicable descriptor. And this has to do with the story I was telling you before about inclusion and diversity in the US, but also because in this kind of literature, when we see, for example, I don't know, like a Mexican compared to I don't know, a Spanish person or like a white from the US compared to a white somewhere else, or an Afro-American compared to a Black South African, these groups are not the same. And they're not the same because race, as we said, is a construct that has exposed these populations to different situations. So you cannot compare these two. These are very local descriptors, and also it's racializing and produces racism. So ultimately, what we want to do is to find new relevant ways of understanding difference. No, and as I said, to create these different categories. We haven't done that yet. What we're doing is to do these kind of workshops also with microbiologists, where we ask them. This is done mainly by Aline. What are the factors that are more important for microbiome research? I put this post-it there, and then they brainstorm and they tell us what is important. What we're trying to do with that is to understand kind of causally what is relevant, and perhaps there find also other ways to stop using these other population descriptors that might be racializing. But also this activity is really useful for their teams, because when they're doing this process, they usually find disagreements. So they can kind of say like, hey, I didn't know you thought fiber was important, why? And they can talk about these implicit assumptions that they have. So it is it's quite interesting, I feel, also for them. What we do here is well, brainstorm and then group the different things in bigger groups. Like, for example, here is host, sex, birth, age, and gene. And there is environment, and so also kind of different groups that make sense together. And then draw like these arrows that are supposed to be causal relations. So this has a cause for that. And then the numbers indicate how big of a cause. So 5 is it causes a lot, and 1 is like it's not so important. So this is a negotiation process that they do in about 1 hour of discussion. And this we will also analyze in order to understand whether there are differences in how human microbiome researchers understand or frame causally what influences the microbiome, depending on when we are going to do our these workshops. So we have done this in Europe, in South Africa, in Mexico, and we will do this soon in Colombia. So we still don't have any results. This is the other thing we did in South Africa. We did this drawing that I think I shared with you. Is a result of a participatory work. Again, we're trying to make them just like, share things and also learn from each other. So we had this at the beginning of the workshop, and everyone had to write something like what excites you about the microbiome. And the artist did this beauty, so that was really cool. We didn't do that ourselves. And this is just an image of the World Cafe, where people were just like discussing while the artist was in the back drawing. And the result was this image, and digitally it looks like this. And to us, this is just like an image, right? But it's also something that at the end, when people look at this result, they were really proud of themselves. And they were like, oh, wow, this is what I said in my contribution when I answered this question. So it was kind of a way, I think, to generate also a relation between themselves, which I think is also important to, eventually make or like realize different ways of talking about human difference. So that's all I had to say for today. I hope that was interesting. I'm very much looking forward to your questions.
Okay, great. I think we can have a 5-minute break. ¿Es un café?
Sí. ¿Te acompaño?
Sí. Ahí interrumpimos.
This map is really interesting. Apparently, it's people that quite generous idea of what white was. A lot of Africa is white. You went to apparently yellow. Map doesn't look very good. To design is too good to be.
Yeah, there's a debate at the like 19.
1909, I don't think that's the date.
Oh, really?
Because I don't know. It just looks the workers. Yeah. It's 1960 or something.
Yeah.
Sorry, I wasn't.
Kind of all the time. It's a lot of stuff from the 20th century.
Okay, great. I think we're all back. Yeah, that's it, yes. Okay. So, questions, comments? Yes, Marco?
Thank you so much for your talk. That really helped. Which use them. So I have two questions. Curiosity. I was wondering, first of all, concerning the reductive methodology that often microbiologists imply. It's kind of, okay, we take the example and we did the job, but so they do not take account of the surrounding. I don't know if we can talk about the environment. And I was thinking if it's relevant here, or what do you think about all the research on exposome that are, it seems to be the totally or the environmental exposure of the individual. If there are some relation, if it's useful for your project, since there are a lot of work already done there.
Yeah.
And the other is that you're talking about is another methodological question. I see how you plan to create this international community. But how do you plan or think or options to do this ethnographic work in science? How you, logically speaking, because it's all the kind of problem. Now we talk about biology, that after that we cannot write. We talk about this. How can we formalize? There are in many ways, but I was wondering what was your plan on this?
Okay, thank you.
Thank you
very much. Yeah, great catch with the exposome. We were recently also in a workshop trying to figure out what similar challenges have, because the exposome is, the total exposure of people to different things, like from air to everything. The microbiome sometimes is considered to be part of the internal exposome by some people. And I don't know, as for how useful it can be, I think both have similar problems, because kind of what you're seeing, and this is also something I think why race is so pervasive, or ethnicity and so on, is that you have so many factors going on, right? You have like everything can somehow have an effect on your microbiomes. So it's really difficult to make sense of that diverse set of factors, and then it's kind of easier to say, oh, well, you come from this place, or you're such and such race, and kind of like reducing the amount of variables. So I think that problem is similar in both. I think also might be interesting to look at whether the exposome has more specific results on what kind of factors have the specific effects. But I think a problem there is also a tendency to molecularizing the social. So, and it's a problem. I mean, in a way, I understand why that happens, but also it's a problem because the exposome, well, depends. There are different ways of doing it, but a lot of them is basically trying to understand what kind of, the relation of the specific factors and biomarkers in your body through identifying the specific molecules. And sometimes in the microbiome, I think also it's not necessarily only about that, especially because in the microbiome, in contrast to exposome, for example, I think you're facing a different kind of complexity where one factor is not always having the same effect because of the functional robustness of microbes. No, it's the case that sometimes, so you have microbiomes with different taxa. It's not the case that always this same combination of taxa will give you the same functionality. You can have other combination very well of taxa and have the same functionality. And this is also because it's an ecosystem, right? So I think could be an indication maybe of how to make measurable the environment to some degree, but I think there's also challenges in that sense. And I don't think we have seen a paper or a publication where microbiologists are trying to use it. It might be interesting, but I don't know.
Okay, there is not an established link.
No, I have
science leader.
No.
Okay.
No, I don't think so. I haven't read any paper that says I will look at exposome or we look at the exposure of. Yeah. But at the same time, I feel that it's also very complex, and looking at the exposome, you also see similar problems in the sense of this technology gap. So you do see also like a overrepresentation of the specific places.
Yeah.
You also see some sort of normativity on what is a healthy space, as in the sense like, Greek, etc. So yeah, I think both are kind of struggling with similar challenges, although they are connected, no, in the sense of the exposure. And also I wanted to say just that I was talking a lot about microbiome in general, but I was mainly talking about gut microbiome. If you look at the vaginal microbiome or the skin microbiome or the lung microbiome, they have other challenges. And they also, at some point, the exposure part might be more relevant. For example, with the skin microbiome, There was a study on the metro, the subway in Mexico City, and that show how kind of people traveling in the subway will share specific microbes in their skin and so just because of the exposure, no? Which is different than the gut, no? Because the gut is more like what we eat and stuff. So it also depends, I guess. And about the methods, so I don't think we're gonna do ethnography because we don't want to talk about microbiologists. Actually, what we want them to do is to think in other categories. So what we're trying to do is rather to, as we did in South Africa, to create spaces where they can reflect a little bit more or have the time to reflect or to express some sort of worries they have, which they have a lot. And also we're trying to also to provide with conceptual help, you know, like with the philosophers can sometimes just have to, you know, bring new perspectives to tackle a problem. So for example, when this distinction between race, racialization and racism, I think that can help them as well. Because many times this debate about race and stuff becomes, but I'm not a racist, right? And of course you're not a racist, or maybe yes, I don't know, but like most of the times you're not a racist. It's rather other things that are producing this reading of your material. So we bring them these definitions, and then you kind of give them a bit more tools to then distinguish what the problem really is. And so we're trying to do that also with the cognitive maps, the quasi-cognitive maps. We still need to analyze those. But another thing we want to do is to just follow up with them into organizing other meetings where we can just talk about different ways of labeling people, like bringing perhaps an option and just see what they think about it. It's good, not probably it's not going to be good, but that's precisely what we want, no? Like kind of the discussion. In our 2 years, it has been really, I mean, it's a kind of also a bias, right? Only you deal with the microbiologists who want to be with you, but these ones have been very, very productive because they're really interested into really finding different ways of talking about groups of people. And as I said, we still don't know the answer. I think the strategy is to just like continue the conversation and try to get a solution together. We are not going to create that for them. It's more, I think it's more something kind of an iterative process of thinking what can work. And of course, there's not going to be one solution. I think that's impossible. It's rather kind of, I think, to generate kind of the reflective practice that you can have, that we have, but as a scientist, probably you don't have time to have. And that can help also as a way you teach to your students, or you do science in your lab and publish stuff. Yeah, we know that it's also very problematic because then you have to publish and then you have to use specific categories. So there's like a lot of troubles going on, but step by step. Yeah. Thank you, Marco.
Yeah, Victoria?
Thank you for the talk. It's really interesting. I had a question because I think I saw on big image
This or the other one? This.
Yeah, and silence non-human voices, and I was wondering, I suppose it's related to like anti-speciesism, but because it's about human microbiome, I was wondering how is it relevant in this case?
Which thing were you saying?
Well, it's a vitality more than
I'm sorry.
speak about the collective when engaging with ethics, human nature, and philosophy. See the other one. I don't remember exactly what happened there.
Okay.
Who are they? What are they doing? I think it's just like because this thing was created during in the workshop. We had different talks. The first one was on antimicrobial resistance. It's over there. And then it kind of started creating. And at the end, we did have a talk on kind of this nature-nurture dichotomy, and also how microbiome research tends to biologize also and not take in consideration microbes as living entities. So I think it's coming from there. In a way, I think the point is also to take this in consideration in order to think on ethics that are less human-centered and more like widely also to non-humans. Also, because if you look at that, it will have an impact on how you take care of your environment and etc. I think it's related to that. Good observation. Thank you.
Thanks a lot for the presentation. This was really interesting. I have a question about, so you presented at some moment that like the databases, for example, are very much skewed towards European, Northern American relations. And I was curious about the research you're talking about, like how big, like you talked about this, but like how much the local scientists are actually participating in the research. And you said that there is obviously sort of technological separation, that they send samples elsewhere, but for example, in the data analysis or writing manuscripts, do you also observe similar kind of dynamics or not?
Yeah, I think what we have observed, this is just observations without methodology. So what we have observed, what we have listened to is that there's a similar gap in the sense that, so yeah, big research institutes are usually in the US or in Europe, and this is related to funding. And there's also a lot of, and there's of course different groups working in microbiome in different other places. But the problem there is like where the money comes from, no? And we have seen a lot of dependency from the US, for example, the Melinda Gates and Bill Gates Foundation, but also the NIH and the Wellcome Trust are quite important. And what happens there is a dynamic where you as a scientist you're not asking, oh, I want to do a project on this. It's rather, oh, there's a grant about this. How do we get it? No? So kind of in a way, yes, there's like people going like working there, but they're kind of also somehow going into the direction that is convenient to have something to work with. And about publishing, so one of the things we also have heard is that this dependency on sequencing. So you have your samples, but you don't have like how to sequence them, or it's very expensive. So you send them over. And what happens then is that sometimes, well, the first thing that happens is that they're sequenced according to a ontology that fits the sequencers, so not your own. And then sometimes they also have the chance to publish before you based on those samples, which is completely unfair because then you're like behind also in publication terms. So this is kind of the things that I think are not only like unfair, but are also central for research and what kind of epistemic questions they can have. No, I hope that answered your question. But we don't have like we didn't do interviews for that. We just got like what we saw in the work cafe, but we have seen with other interactions.
I have a question about that. So if I understand correctly, you also observe that their epistemic freedom, if you want to call it, is limited by the almost relationship of patronage that they have with the West. But I was thinking whether, and this is perhaps the bigger question, whether they are thinking of race in different ways, even if slightly, and sort of if so, how? So is there epistemic difference between how a South African microbiome research would think about race than let's say a European scientist, and sort of yes. So my main question is, what's the alternative label to address genetic diversity or diversity in general that is not race-based? I guess that's the bigger question, and then I think that for me also relates is sort of, is there any talk about sort of somehow data-driven approaches to analyze some of this diversity? Because, for example, if you put up the panel where there was all the different sort of things that affect the microbiome, where with the post-its.
Yeah, here
Yeah, this. So I saw this and I was thinking, well, I thought first of all, it's very interesting. And I have a question. Why did you, if you can talk us about the negotiation process, you said that they were negotiating, and I was looking at this and it reminded me a lot of sort of this sort of old school systems thinking in systems biology, where you sort of have this sort of machine, right? That sort of there's different effects, and then the idea will be to control. And I'm wondering whether statistics could replace the negotiation process and whether that's a good thing or not. And whether there's discussions about, well, we are not talking about race and categories, they are sort of just categories that emerge from the data, and whether that's even a bigger risk to racializing practices, because you would say, well, I'm sort of, this is theory less, and it's just sort of data-driven racism, basically.
Interesting. I haven't thought about the data-driven racism so far, but let me think. So the question, what will be the answer? I don't know. This is we're gonna analyze not with machine learning or anything, just like gonna try to analyze it as matrix and so. And this is more related to kind of as a previous step to defining categories that they should use. Is more like to understand, okay, in your way of seeing what is important, and try to identify then what is important, and then we were gonna ask them, is this really what you think it's important? So kind of in a way to not let it into like a AI driven, try to generate it ourselves. Probably it's also something that will happen. But you have a different question at the beginning.
Sorry, yeah, I was a bit confused by myself. Yes, so whether the South African, how do they think, and whether this, this.
Yeah, no, it's completely different, and you can also see, yeah, exactly. That's exactly what we have seen. That so if you're like dealing with your own local population, you know which variables in a way are important. South Africa, as you know, had apartheid, and that really kind of structure society in very important ways. And still categories that are used by broadly, and that are not only biological, or I would say not biological, but rather socially, culturally embedded into ways of living of people. And that's completely different than when here, how we understand, we don't even use race, probably, but we still see differences. Or the US or Latin America, no? In where we also don't use this category. So yeah, there's local understandings of difference. And this is also why we're not saying, oh no, eliminate race forever, because maybe it's useful in some context. So kind of the point is to find a way to make those relevant things visible and kind of be able to publish, for example, using those labels. So the problem is really big, because actually you have all these databases that are already labeled in a way, in a different way. And usually there's like white European or whatever, no? So it is really difficult to come out with a new way of categorizing people. And this is also for this data, historical data that you have, the infrastructure of databases, or just simply how technology works and how it is set to work, no? Also, you want to make your research comparable. And if you don't have comparable labelings, then it's probably very difficult to make this comparison. So these are the problems that we have. But there's also like, I think, a possibility of trying to at least make the problem visible, because as it is now, sometimes it's assumed that these categories are interchangeable. So it's kind of not explicitly say that there's a problem, but there is. And I think at least that will be a first step to say like, okay, it is never comparable when I talk about white in the US and white from South Africa, because they are very different kind of groups. And at least frame the question like, in which ways are they comparable then? Just because they're auto-defined as white, or because their skin is white, because they're rich, because they live So kind of what I think will be an interesting way to go is also to look more into what Marco was kind of suggesting, like these relevant factors that can shape a population as in where they are living. Like, for example, if you live next in a mountain, maybe you have similar factors kind of shaping your health or so, in contrast to people living at the sea, or things like that. Not so kind of like are things that might be comparable between different people living at the seashore, in contrast to people living in the mountain, instead of looking at white people everywhere, you know. But yeah, I don't know. It's a big problem. And we're gonna find a solution. Can't know what.
I have more questions too. So Marco, yeah.
Any other? So thank you much also for this whole answers. I don't know, maybe it's a question a bit a controversial question. I don't know, maybe it's just confusing. The junction between race and microbiome, no, is there that you want to. So you say, okay, we try to make a community, to talk each other, to find out new categories or label, to investigate microbiome in order to propose new way to characterize people, right? It's a little bit one of the activities here. And my question, I don't know, maybe it's not clever, but my question is, there is some kind of trust in science, say that if we trust microbiologists from different parts of the world, we hopefully can find a new way to characterize that less rational than the research that you proposed us in 2015. And so there is an assumption of trust, or no less. There is something that why you think that I agree with you, but I'm just curious about what is there. I don't know if it's the normative part to say, okay, with this research we will be able to reduce this bias, and but it's science, it's kind of because we trust science, it's like that. I don't know. This is
yeah, that's a very good point. No, of course. Like it can be also a very big critique, right? Like, why are you giving more power or like more relevance to the people who has with their work have created scientific racism or so?
Sorry, do you want to also work on the more ethical parts, or only on scientific discussion? So I didn't understand if you want to get power also from work on ethics, for example, bioethics, and then gather together with the scientific part. Say we get more power because we create a more interdisciplinary approach to this. Maybe this is a curiosity.
So I think yes, I think so. The reason why I want to work with scientists is because I think they have the power to create reality in a way.
I agree.
And in a way, I do trust that they are going to do it because they like science. So I kind of do have this trust in them. But yeah, you're also kind of feeding the power machine, right? So, but then what I do see is that when, as I said, I also have like a bias because the people I'm working with is the people who are interested, who already know that race is wrong and they want to do something different. So the other ones, they don't answer my emails, right? And they are too busy doing other stuff. They are there. That's okay. If you don't want to talk to me. But then I'm going to talk with these other guys and try to build like a bit of a critical mass from there to the degree that is possible. And then the ethics part, the ethic element is always there because when we're talking with scientists, that they don't necessarily think that the research is wrong, as I'm claiming. This is something I want to convince them because then if you know that your research is wrong, then maybe you want to change it, not just because it's wrong ethically. What they usually kind of perceive this as ethically wrong. So kind of the reflection on the ethics part is very fundamental. What I want is kind of to show how this ethic part is not like additional. It's actually at the core of also the epistemic questions. So that's kind of what I guess the philosophical work we have to do to kind of frame it that way. But of course, I do see the worry, and I have been asked also in the past, why the scientists? Why not go into the communities? The communities at the end are the ones that are being studied, objectified, sampled, etc. And yeah, I guess that would be a different kind of research. But I do think that if the scientists are the ones who are doing that, maybe you should go and talk to them in order to kind of change the practice. We will see. Maybe my next project is on the community work or something. I don't know. Yeah, thank you, Mark.
Thank you.
I have a follow-up question. Maybe it's the same question. So if you say it's the same question, same answer, it's also okay.
Okay.
But I was also having a similar worry. For me, I was thinking in terms of like, I do see that there's sort of epistemic consequence to, let's say, an ethical or political issue. But I'm wondering whether reforming epistemic means is the way to addressing the political issue. So in the sense of like, well, at the beginning you showed this, basically the history of race thinking in science, and it feels like always racists are able to sort of turn the whole thing around, right? You have like, I mean, I remember when I was growing up, one of the first stickers I put in my computer was this thing that said, there's no races, there's only one race, the human race, and sort of this sort of 99%. But then that was turned, like you said, also in the genealogical testing, where you could sort of see where you come from, where sort of they exploited the critique of the orthodoxy of sameness, right? So it's always this they are sort of racist, not scientists, but the system sort of always is able to exploit those things. Yeah. Or for example, I mean, just to bring the point very concretely, in Ghent, there's this philosopher, Nathan Kofnas. I don't know if you know about him. Well, he's a racist philosopher and talks about race realism. And so his point is that, well, we should stop race mixing because we need to protect the diversity of each race. And so this comes with sort of certain political, and this is just around the corner. And so my question is whether sort of, I don't think that conversation with this kind of person is enough. I don't think that even having a critical mass of sort of reflexive scientists is enough. I think in this specific case, the strategy would be to isolate these people as much as possible and to decrease the amount of power that they have. So by not having access to university networks, legitimacy, and so on. Yeah. So I'm just worrying about that.
Yeah, no, you're completely right. Oh my God, this is okay. No, you're right, and I think so. I don't think we are gonna solve the whole problem of racism, especially in current times, where I mean the whole debate on race in the US has turned around, where you're not able to talk about it in biomedicine, and then it's like, yeah. Anyways, so yeah, there's a lot of white supremacy going on in the big spheres, and it's becoming very popular. And I don't think my project is going to solve that, but I think we can continue trying to build a lead. I mean, I do think that the critical mass building is an exercise that can reproduce itself. And I think it needs a combination of efforts. So yes, it needs universities that don't hire these people, and it also journals that don't publish these people, or like that you can reject or retract publications, but like, I mean, currently, honestly, I see the world going in the opposite direction in terms of racism, sadly. So this is why I continue doing my talk on race. of them, but like it's just like surprising that these kind of arguments. I mean, it's not even like very informing genetic terms. Like
I don't know.
I don't think so. Isolation is never. We have never. I mean, if you look at the history of how people have migrated in time, there's no way you can identify individual groups of people that were all coming from the same place.
Yeah, but what I worry is that, for example, he's using this notion of, oh, there's some sort of biocultural diversity of like it's genetic and cultural, and we should protect the diversity. And of course, this comes with the whole replationist agenda, the theory, replace theory. And so
What's his name?
Nathan Kofnas, and he's being hired like this week's.
Is that the problem you were mentioning before?
Yeah.
Okay. Yeah, no, I think it's more like, I mean, of course, like racism is also an institution, and I think we just have to attack it from many different ways. Because the one I have been concerned in my research for long is like the one that comes from science. So I don't want biologists to be kind of telling again the same story that because it's not true also. So yeah, but I think it's much more bigger than that. It's why race is so successful, right? It's so entrenched in the way we see things that it's very difficult to avoid. I'm very surprised about these things.
No.
Very interesting.
I just have one more question, sorry, very specific. But has there been any critique of when at some point you show the three stages of
Yeah.
the pre-Neolithic and I mean, of course, the epistemic issue is obvious, right? They haven't been able to time travel to the Neolithic era and or to the agricultural pre-industrial era, so they are just assuming like this is like that era.
Exactly.
But is there been any within the community any pushback against just the epistemic stuff about that?
Like within the community of microbiologists, I don't think so. It's really at the core. It's really like you assume that this is how it is because it's linked to how diverse your microbiome is. So there's a measurement of diversity, which is very complex in itself and problematic. That is linked to these different, to this framing. So there has been a lot of critique from us, not from the anthropologists and the historians and all these people who say like, hey guys, like you cannot do that. But this is still very, very much at the core of, and this is why it's problematic. If you see how science papers are written, well, also our papers, you always start with a literature review, no? And say, ask this, this, and they're like very core papers that have always kind of been lined up there as like kind of the analytical, the framework that then sustains or justifies why you're doing the study you're doing.
Mm-hmm.
So I think this is also something that we're trying to kind of reflect with the people, like why are you using this? In a way, it's also, I think it would be useful to talk of this problem like the critiques to evolutionary psychology, which is also like, hey, you don't have a possibility to go to that time to see how was that working out? No, you're just like inferring from what you see today, which is kind of a similar, but I haven't seen anything from the field.
So there is no sort of science for the people, like scientists, like what's his name? What was the geneticist? Sorry, Lewontin. Yeah. So back in the day when sort of Lewontin and Levins, they were from very much within criticizing the genetic reductionist ideals, but I'm wondering if those kind of, we've lost the Lewontins and Levins and there's just no more.
Yeah, I haven't seen anyone, maybe soon.
Yeah, I do also have a question regarding. You said that, like, when you started this work, one of the things you were expecting to see, and you observed it to a degree, is that there will be these sort of, I don't know, scientists coming from rich developed countries trying to solve their problems and sort of studying local populations just to solve these problems. And I was just curious, like, to which degree you observe this sort of actually as sort of, it's connected to my previous question, to sort of external questioning, and to which degree it was motivated also by the problems of local scientists, because I mean, most of the developing countries are already urbanized or very rapidly urbanizing right now. So it seems to me that they must have similar problems as sort of Western societies have, or maybe even similar problems, but aggravated by the fact that they're not as rich as Western countries. Whether there is sort of internal motivation, for example, if you go, I don't know, to Brazil, of people saying like, okay, we in São Paulo, we have these horrible problems of urbanized societies. We should go and see how these people back to nature live. And
yeah, that's interesting. I haven't seen it in South Africa, perhaps you see this, and then they rely on the notion of transition. So they say that different populations and different, like in transition, and then they study how, for example, people living, so this study was done in Johannesburg, so it's like Johannesburg, and then the different kind of townships outside. So they see how people live here, but these people go every day to work in the city, and then they come back. So they're trying to kind of figure out how these two things are working. And I would think that this is more or less more like a local kind of question, because their focus is to understand also how these local people who are living here are changing their microbiome because of going to the city. But I don't see this as being very often the case. So, for example, in Sao Paulo, that's not how it works, or in Mexico neither. No, it's more, I don't think it's necessarily that specific, the kind of question they have. But I have also seen in particular in this, by these researchers also, and also working in Johannesburg, is that they've really tried to, like, very specific, these questionnaires, like dietary questionnaires, so that really kind of can grasp the local foods that people most regularly will use. So they will kind of shift the questions. Instead of kefir, they will use, I don't know, something with green and so on, to try to make it more local. So that's happening, but it's not really widespread. And then what happens there also is that you have to, I guess it's all about how big the different labs are and where do your money come from also. Again, in this particular case, I think these studies were also very much done in collaboration with people also in the US, but perhaps with more like a framing on, let's just not figure out our own diseases, but so yeah, that's a good thing. Yeah.
I'm good in French, but not in English. I translate my questions and I post it.
Yes, so I'll read his questions.
Okay.
Well, I put the first two together because I think they are the same package. Yes. Yeah, they're similar. Would you consider your work to be part of a decolonial approach? Because it seems that your research aligns with this broader decolonial trend across several scientific fields, particularly through the critical examination of biases, including racial ones in this case. And would you agree with this interpretation? And yeah, where do you see yourself in the decolonial approach?
Yeah, I think it would be part of that. Sometimes I like the label of the colonial because I think it became already very colonial. So I think it aligns with those objectives on acknowledging colonial ties and how colonialism has shaped to a large degree the amount of money and power that different regions have. Yeah, but I don't like sometimes very much the colonial debate because I think it just like can become a bit like an empty label sometimes, and then you just say like, I'm decolonizing, but you're just like kind of, I don't know. But yes.
get this one. So it seems that your work both highlights the existence of biases, like you said, and proposes certain methodological avenues to address them. And where do you put more your focus on bringing those biases to the light, or on methodologies to precisely avoid those biases?
Interesting. I think both, but maybe I guess like in the process of how things happen, we have first tried to identify which biases are those in order to then try to, with the methodology, kind of change them. But I think also what we're trying to do is also to look at our biases, right, as scientists, because of course I have a stand on race, and I of course have like an objective, and that can also, I guess that also plays a role in the research I'm doing. So we're trying to also try to kind of have an interactive process of looking at our own biases and their biases, and then try to also find ways to solve them or making them explicit. I do trust that making explicit something can help a lot, because then you know at least that is there, and that something needs to be done about that. I hope that helps. Thank you.
Peter?
I have a question, but it's not really about your research, so I'm sorry if you don't have a question. It's about the microbiome itself. I don't know much about it, but and I'm kind of struck by how romantic these feel. I mean, romantic in a bad sense of the word. In the sense that, I mean, why would you think that? I mean, it seems very unlikely that these primitive people, like that would be sort of more diverse always. Like it seems very like this back to the when everything was good and so on, and now industrialized societies and it's all there's no diversity anymore. You're far from nature, and I don't know. It feels like a very unscientific sort of way of looking at it, yeah, it seems like it, certainly rich cities or rich communities, there might be access to much more diverse foods, while in very poor, very non-industrialized places, there might be access to a very, very limited amount of food, while in Sao Paulo, in poor environments, maybe you do get a very like, I cannot get my question exactly straight, but I was wondering how this picture, this ideal of the less industrialized places having more diverse biome, microbiome, and that also would be health beneficial, how substantiated that is by evidence, or whether there's a lot of real romantic feeling behind it.
Yeah, that's a really good question. So I hope my PhD research no, but so I think there's like so there are very important papers, publications in the 2000s that start showing or that start sampling like the HATSA and groups like this, and they start like saying, oh, they have like a larger diversity, micro alpha diversity than the comparison of the US or whatever, no? And I think in that sense, okay, it could be the case that the diversity in those groups at that particular moment in time where the sample was taken was bigger, the alpha diversity. And it was a finding, right? And then kind of, I think what happened is that started come together. So like these kind of groups have bigger alpha diversity. Another set of insight was, oh, bigger diversity, and specifically the presence of this and that taxa helps with digestion, or the opposite. The absence of this and that taxa that we found in this group kind of explains that you have colorectal cancer, no? So then comes the comparison. Okay, this group has this taxa and more diversity, and this group seems to have less.
Mhm.
And then comes the contrast.
Mhm.
So I think it's a combination of things. So on the one hand, there's studies. I mean, they did have samples, and you read at these papers, and there's like a lot of statistics going on, a lot of things to say about that too. But they have samples, statistics, and then narratives that then I think at some point got just like really reified in the sense that we just assume that this is the case, that there's a difference in alpha diversity, and that the presence or absence of specific taxa are related to specific problems. I don't know how much I don't know how rather, this came to resemble so much the noble savage kind of vision that somehow seems to be there. The stage narrative does remember a lot to cultural anthropology or ethnology, differentiation between the barbarians and the civilized, these things that are no longer used also in those fields. There's also a lot of, I think at some point, at least in anthropology, the idea of the value of the savage anthropology that is started with Boas, for example, where you have to go to these places, to these indigenous communities that have so much knowledge, and it's going to disappear. And so also that is playing a role. Mm-hmm. But I honestly don't know. I don't think microbiologists were thinking, oh yeah, Boas and Tyler. I mean, I don't think it comes from specific sources. I think it's maybe a way of thinking about this contrast that we have. I like a lot a study paper from Stuart Hall that is called The West and the Rest. And because it kind of, and then there's similar things like this essay, but this is, I think, really interesting because it kind of just like names how often, how used we are to this contrast between very big idealizations, like the West, that's something that exists, well, whatever that is. I mean, Europe and everything's so diverse, we can live in very different circumstances. As you say, like rich areas might have like all the probiotics in the world, etc., and very poor areas have nothing. So kind of more like the logic of opposition between an idealization and the other. So kind of, I don't know how it came to be. I have hunches, which I just try to explain, like kind of a combination between studies and then kind of bigger ideological kind of things we are embedded in because we have some sort of common sense of understanding things also. And then now I think the story, what anthropologists and historians were trying to do is to disentangle this, but it's different to say how it came to be, right? Because that's kind of how it was born. Well, I don't know, maybe one of these microbiologists did cultural anthropology and thought about that. That's, I think, an empirical question.
There must be a commercial aspect. It seems like it's very attractive for advertisements if you have your product, your bacteria containing foods, being the one things, and also the indigenous people and the Amazon, blah blah blah. This kind of romantic feelings, people like that in the Western world. So that could. I mean, I would be afraid that this would create a strong bias that even if maybe the microbiologists themselves are not thinking like that, the research that gets sponsored could become useful for that sort of products. Yeah. And so the commercial goals could thereby creep into the research stuff or. Yeah.
No, I completely agree. The field is quite diverse, no? I don't think everyone is. They're very reflective people in the field and everything, but they're also very non-reflective. And there was a very big case also in microbiology some years ago of a microbiologist who was doing products like the bar, the nutrition bar of the that will help your microbiome to become. So yes, that has happened, And it's also something that we're trying also to see how much of this, there's a lot of promises going on, right? Like, if you do this, then like this research will help to do this and that, that can then lead to perhaps commercializations. But I think in our conversations with microbiologists, at least the ones that want to talk with me, they are very aware that this is still very far away from whatever they know because it's so complex. So, but yeah, that's has happened. Also, there's like a lot of at least this person was also implicated in kind of horrible, unethical treatment with populations. So there's all sorts of people. That one was a very famous case. Not everyone is like that, but unfortunately, yes, that can sell very well. And I think it's also important to say that the field is currently quite like really thinking about ethics, especially because they're always kind of in connection with indigenous communities or minority groups. And now kind of they are at least publishing about that, like we should do this and that. Now what we want to know is how much of that is actually kind of done instead of just like said as a common sense. But yeah, definitely it's something that happens and is. There are other fields, for example, like the gut-brain axis that have a lot of promises as well. Like, with diet, you can perhaps cure ADHD or understand, I don't know. Yeah.
Is there an overlap between the gut microbiome community and the nutrition community? Because with that question, I was thinking a lot the sort of this huge debate between vegan or plant-based science versus the Paleolithic meat. Yeah, sorry. But and I mean I know it that at least on YouTube they fight a lot. But about sort of the microbiome that sort of the Paleolithic scientists, well, not Paleolithic diet scientists, argue that meat only is really good for microbiome, and then the plant-based scientists. And I wonder whether those are just two different communities or whether there's some sort of overlap in so investigating the effects of diet on microbiome and this kind of stuff.
Yeah, I think there are many different communities talking about the microbiome. I don't know, that will be my first answer, but what I have seen also is that when in these studies, in many cases, they talk about, for example, the Western diet or the Mediterranean diet or I don't know if the Paleolithic diet, but like these kind of things. But what these big ideas reduce to in the lab is to sugar and fat. So if you're talking about, I gave a Western diet to my mice, they're just giving like drops of sugar and fat. And but I mean, also because you cannot give them a hamburger or something. It's just like, you just give them like the elements that you think are what the bacteria, because the kind of the relation between the bacteria and what you eat is what the bacteria can do with it, right? So if there's a lot of fiber, you need bacteria that do fiber. If there's a lot of fat, you need bacteria to do. So in this case, I would think that if there are studies about Paleolithic something, you should look at the study and see what actually was used in that study to say that it's a Paleolithic. I don't know what seeds or I don't know what they use. But
normally Paleolithic is sometimes they just eat meat basically.
Okay. So this is also something that we were like when you talk about Western, it's just like, what is this really happening when you're doing the study? And really, sometimes it's just like not as Western, whatever. It's just like maybe sugar. Sugar is a problem. But I don't know.
Okay,
but it's interesting. I would like to see those debates.
Yeah, well, you can you won't end it.
Yeah, I guess something also interesting is that together it's related to diet, is that as you see how diet changes because everything is just like more centralized and like, this is another claim that is in like together with the disappearance of these microbes, because you don't have your usual sources of food, then you have to change to supermarket food, then microbiome changing as well. So it does have a connection with that, but haven't seen that
Okay, thanks.
very closely.
Okay, and I think if there's no burning questions, then we will close the seminar. Thank you so much.